Okay, yeah, thank you. It's very nice to have this opportunity to talk about the project I'm currently working on. I'm Kai, I'm from Michigan, and uh, so the method is called matrix completion and coupled matrix completion. And for here, I will talk about the method first, and then talk about the R package. So for environmental chemical exposures, it's like all of us like exposed to all of kind of like environmental chemicals, and based on previous study, it's like 2,000 new chemicals are released to our environment every year in US. It's like 7 new chemicals per day. And to do a comprehensive toxicity assessment of all the chemicals is impossible because in PubChem, there's already over 1 million registered chemicals. And but the related biological data is only like 10%. So yeah, so that's why we need to do some like a computational task, a computational method to predict some targets, then to do some like in vitro, in vivo validations. So previously, computational toxicology method, it's like the read across and the SAR or QSAR or other machine learning method. So all of those methods is actually really focused on the chemical part, which is like use the chemical structure to predict whether this chemical can cause cancer or not, or is there any like a certain marker genes or pathways can be activated by those chemicals. So it's kind of like it's heavily focused on chemical, not like really focused on the biology. So for here, we used a matrix completion method. So usually, so this type of method is kind of like very popular in the drug target prediction. And the matrix completion is like the method to finding the missing values from the partially observed matrix. And for drug target interaction prediction, usually the matrix is a binary matrix because drug is designed to binding like a specific gene or protein. And usually it's like the physical interactions between the drug and targets. But for here, because environmental chemicals, they are not like designed to target any genes. So we didn't use a binary value here. So the method is actually called a coupled matrix completion. And if you want to see the detailed mathematical description, which is in that paper, yeah. And the method is like the input have 3 matrices. So the main matrix contains the interaction values. So the row can be gene, the column can be chemical. And the other 2 matrix, it's the similarity of the genes and similarity of the chemicals. And all of those computational toxicology methods, they have the same hypothesis, which is like a similar chemicals can target probably similar genes. Yeah, so here is also the hypothesis. So this method is different from other previously matrix completion method used in drug target prediction. Is like we directly used the similarity value into the calculation to finding the missing values. And for the data we used for now, it's actually from this database. It's called the comparative toxicogenomics database. So this is kind of like the best toxicology database. And from this database, we download the human data. And after we're filtering some drugs, so there are more than 4,000 chemicals and more than 20,000 human genes. And the existing chemical gene interaction number is there. So it's like a little tiny portion of the huge matrix is existing. Yeah. So the data is very sparse. That's why we need to use the coupled matrix to finding the missing values. Yeah. So this is the data for the main value. What we did is like we actually download the data from the CTD database. They provided the data to have like the chemical gene pairs. So we just count the chemical gene pairs and then normalize that value into a range between 0 to 1. So a bigger number means there's a high interaction between the chemical and gene. So for the chemical similarities, we actually use 2 different methods. All of it is use RDK. So that is a Python package to calculate the chemical similarities. We use Morgan fingerprints and the topology torsion fingerprints. to calculate the chemical similarity. So we have like two types of the chemical similarity. And for the gene similarities, we use the R package, which is called the GLBIO sampling. So this package is calculating the gene similarities based on the Go term they are assigned. So it is not based on the sequence. So because we have like three types of the Go terms, so Go like a Go biological process, molecular function, cellular components, we have three type of the gene similarity. So for all of those values, they are in the region between 0 to 1. Yeah. So then, so for now, this method, the input for this method is only grab the main matrix and one type of the chemical similarity, one type of the gene similarity, then can find the missing values. So for the performance comparison, we compared our method to three previously published methods, which is called the CMF, GRMF, and WGRMF. All of those three methods, it's developed for drug target predictions, and they are all in MATLAB. So I implement the MMC in C++. And for all of those three methods, they actually have WKNK, which is a pre-processed method can be improve the performance of other three methods. So in total, we compare our method to kind of like a 6 method. Yeah, and so then we calculate the AUC for the performance. So for the AUC, how we because for our data is not binary data, so to define the kind of like a true positive and true negative, what we did is like we choose the top 20 very well studied chemical, and then choose the target genes of those chemicals. For the true negative data set, we also choose those top 20 well studied chemicals, and then choose the genes that never reported as a target gene for them. So then we have the true negative and true positive. Then we calculate the AUCs of those data. Yeah, so for now we generate three sizes of the matrices. 200 chemicals by 400 genes, 400 chemicals by 600 genes, and 600 by 800 genes. And the right line here is the AUC of our CMMC method. And from this figure, you can see with the WKNK, which is the lower three plot, you can see the performance of other three methods is improved a little bit. And but our method is kind of stable for all of the different sizes of the matrices. And yeah, so then we performed a case study for our method. So we choose the 18 different chemicals here. And for the BPA here, actually, BPA and its analog, it's the chemical used to generate plastics. Like if you bought the bottled water from Costco, you can see the label no BPA. BPA is kind of banned like 10 years ago in the US, but still have some analogs. So yeah, for the PFAS, it's a chemical used to use their non-sticky pen. So if you go to Target, you can see some products that say no BPA, no PFAS. But there are hundreds of PFAS, so they use different types of PFAS on that. Yeah, for the toxins here, actually the TCDD, it's a chemical weapon during the Vietnam War. Yeah, and the PCB is other chemicals used to produce like a electric device. Another two like a very common compounds. For those chemicals, what we did is like we want to mimic a real life scenario, which is like we removed all the existing interactions of each of those chemicals in the huge big data here, like 5,000 chemicals and 10,000 genes. And to make sure it's a roundable, because those 500 by 10,000 is like a huge. So we break down those the big matrix into small matrices, like 200 data sets. And for each of them, we just remove the existing values, and then we run the 200 data sets like one by one. Then we average the result. Yeah. So after that, we compared our result to CTD, which is the database that we use for the data, and the Compost. Compost is the EPA database, which is independent to our data. Yeah. So for this figure here, we just compare, because our result, it's not like a binary, so it's like a value. So we just sort the value from large to small, and then to see, okay, how many of the top predicted genes in our result is also the top target genes in CTD. So we can see for those 6 chemicals, especially for the BPA. So it's over 80 genes, top genes from our predicted result. It's also top genes in CDT. And for the COMTOX here, it's similar. Yes, here it's similar. So it's a percentage like how many top target gene from our result is also top target genes. Top target genes from the COMTOX. And then we also compared our result to the AC50. So AC50 is a concentration of the chemical can cause like 50% of maximum activity of that gene. So for those 1, 2, 3, 4, it's like 11 chemicals, they have the AC50s on the COMTOX, and we compared that. So a lower AC50 values means a high interaction between the chemical and genes, and our result is kind of like a match. So in our result, a bigger number which is have a lower AC50. Yeah, and then for the BPA, so in our result, we found this GPR ER1 gene. It's only a target gene in for BPB in the case study, and yeah, this paper support our like a result, which is like the BPB have a 9-folding binding affinity than BPA, and yeah. So for now, everything is for those study. The code is in C++, and then I'm working on our version of the method, because R is more easily to use. So I use RCPP for the CMMC, and then they have RRDK version, and the Ghost assembly is already in R. So yeah, so everything is in R now. And for the design, like the user can just provide a single chemical or a chemical list. So our package will provide a predefined gene similarity and chemical similarity. Then just run R code to generate the overall result. So I'm still working on the R version. Yeah. So the core function is already finished. Then I'm working on those functions for validation of the results. And for the runtime comparison, so the C++ version is like it's only take like a second for one run. Yeah, for one calculation. But I run that on a Linux server. Then I test the RCPP on my own laptop. So it's a little bit slower, but it's still acceptable. It's only like a second, half a minute for the biggest matrix. And for future plans, for now, it's a CMMC. It's only taking 3 matrices into the calculation. For future plans, like because we have different types of the gene similarity, different types of chemical similarity, so we can use the so the second one is called a couple of tensor matrix computation. Then we can also expand the matrix, the main matrix, into a tensor to contain different, or like a tissue specific interaction. Then they call a couple of tensor completion. Yeah, basically that's all. And I would like to thank my wonderful advisor, Maureen Sutter, Dina Dornan, and Justa Colacino, and also this support is mainly supported by my Kin and I grant, and also the other grants. And yeah.